Hello there, A&P students, and welcome back. We have been looking at one of the special senses, vision, and we will finish up that discussion with this video. We will be studying two other special senses, and those are hearing and vestibular system. And then we'll finish up our semester with a very brief introduction to the autonomic nervous system. Phototransduction is the process by which light energy is converted into a graded potential. Photoreceptors are the rods and the cones. They are modified neurons that are sensitive to light, and they contain photopigments that change shape as they absorb light. Recall that rods are more sensitive to light, so they function well in dimly lit areas. However, they are low acuity. We are able to see at night because of rods, but we do not see a sharp image. Peripheral vision utilizes mainly rods, which is why it is difficult to make out details when we're not looking directly at an object. Our peripheral vision is best suited for the detection of shapes and movements so that we can detect danger. Cones, on the other hand, are less sensitive to light and rely on a greater intensity of light to work. Therefore, they do not work well in low light conditions. However, the trade-off is a much sharper image that is in color. Let's take a look at the reason behind this difference in acuity between rods and cones. Rods participate in what is considered converging pathway. This is where a large number of rods stimulate a single ganglion cell. And that ganglion cell is receiving information from a large area of the retina. Cones, on the other hand, have a one-to-one relationship with ganglion cells, and they're said to have their own labeled line into the visual centers. A ganglion cell will receive information from a much smaller area of the retina. To make it easier to understand why this makes a difference with acuity, think of the pixels that make up a photographic image. The smaller the pixels and the higher the number, the sharper the image. Our study of phototransduction will only be a skimming of the surface of this very intricate and detailed process. For our purposes, we need to understand that photoreceptors absorb light, and they cause an electrical change in the membrane of the receptors, and this change is what begins the conversion of light energy into an image. Basically, the photoreceptor will either depolarize or hyperpolarize depending on light conditions, and these changes in membrane potential are graded, and they're known as receptor potentials. The photoreceptor will cause an effect on the next cell in line, which is the bipolar neuron. Bipolar neurons will also develop graded potentials, and they synapse on the ganglion cell. It is the ganglion cells that develop the action potentials, and depending on the conditions of light, they will either be active or quiet. In conditions of light, the ganglion cells will be firing, and the brain will be producing an image. The axons of the ganglion cells exit the eyeball at the optic nerve, and at the chiasm, a number of those fibers will cross over to the contralateral side and join the ipsilateral fibers, coming from the opposite eye. The fibers synapse with other neurons that will project to the primary visual cortex in the occipital lobe. The visual fields from both eyes overlap considerably. However, each image is slightly different. The brain will fuse these two images into one, giving us depth perception or 3D vision. Now let's take a look at the ways that our eyes accommodate for changes in light intensity and distance. We will first look at how the diameter of the pupil changes with differences in the light intensity. Since light is the key to vision, there needs to be a way to control how much light enters the eyeball. Too little light and our cones are not functional. Too much light can be damaging to the sensitive photoreceptors. The amount of light entering the eye is controlled by the smooth muscles of the iris, and these muscles are controlled by the autonomic nervous system. We have the pupillary constrictor, which functions as a sphincter, and the pupillary dilator, whose fibers run radially. In well-lit conditions, the sphincter contracts and it closes down the diameter of the pupil, allowing less light to enter the eye. Pupillary constriction is controlled by the parasympathetic system. In conditions of low light, the pupillary dilator will contract, increasing the size of the pupil, allowing more light to enter the eye, and this is controlled by the sympathetic nervous system. Let's take a look at a video that tests the pupillary reflex. In this video, the doctor is testing for the direct or ipsilateral reflex, as well as the indirect or consensual response, which happens on the contralateral side. You will practice this in the laboratory.
Hi, I'm Dr. Chuck Maxner, one of the neuro-ophthalmologists based here in Halifax. And what we're going to demonstrate for you today is checking for the pupil function to light in preparation for understanding the afferent pupillary defect. So when examining the pupils, the key point is to have the patient in a somewhat dimly lit illumination to enable the pupils to dilate slightly so that you'll be able to pick up the pupil response to light. It's always important to realize that because of the balance of the neuroanatomy controlling pupil function, that when you stimulate one eye with light, both pupils should constrict. So you give the person a distance fixation target, so if you just look at the distance target, and then you dimly illuminate the pupils to see that they're symmetric and there do not appear to be any structural abnormalities. And then you shine the light in one eye, and what you should see in that eye is a brisk constriction of the pupil. And if you're able to see the opposite eye at the same time, there'll be a consensual response, and that should be symmetric. Then you can come across to the opposite eye, and same thing. You can illuminate the pupil, it should constrict, and the contralateral pupil should constrict just as well.
Thank you, Dr. Maxner, for that great demonstration of the pupillary light reflex. Let us look once more at the path that light takes as it enters the eye. The light rays will first come upon the cornea and pass through this structure. And as it does, any rays not perpendicular to the cornea will be bent. They will then pass through the aqueous humor and enter the eye through the pupil. And these rays will pass through the lens. And again, if they are not perpendicular, they will be bent twice more. The rays then will pass through the vitreous humor on their way to the retina. As they approach the retina, they will travel through the fibers of the ganglion cells, passing by the ganglion cells and the bipolar cells to be absorbed by the photoreceptors. Recall that the job of the extrinsic eye muscles is to move the eyeball so that light rays reflected off of the object of interest are directed toward the fovea centralis. When we are gazing at a distant object, our eyes are more relaxed, and this is a more natural state. As a matter of fact, if we are reading or doing close-up work for a long period of time, and we begin to develop eye strain, the best thing to do is to look away at a distant object. You will immediately feel a sense of relief. Try it right now and feel the difference. I'll be here when you get back. But make sure you pause the video. Could you feel the difference? Well, you might be asking yourself, what is changing in our eyes that causes this difference? As we look at a distant object, the object appears small, and the light rays are reflected off of it in all different directions. Our pupils are more dilated to allow more light to enter our eyes, and the rays coming from a distant object will be nearly perpendicular to the cornea, as opposed to divergent and convergent rays. Because these rays are more perpendicular, we need less refraction to focus the rays at the fovea centralis, and the lens is in a more relaxed state. Whereas when focusing on an object that is close up, the light rays reflected from that object are diverging or converging onto our cornea. We need greater refraction to focus those rays at the back of our eye, and the lens will be thickened. Let us look at the ciliary muscles to understand how this is accomplished. The ciliary muscle, which is part of the ciliary body, is a sphincter-like muscle that is attached to the suspensory ligaments that attach to the lens. The lens is a flexible, transparent, biconvex disc that can flatten and become thinner when it is pulled on. With the ciliary muscle relaxed, the suspensory ligaments are taut and the lens is flattened. And this is what happens when we are focusing our gaze at a distant object. When we change our gaze to focus on a near object, the ciliary muscle contracts, And the suspensory ligaments let go and loosen, and the lens is released into a thickened disc, which allows for greater refraction. As we age, the lens loses its flexibility and we become less able to focus for near vision. This is called presbyopia. And so, to summarize, as we change our focus on near or close-up vision, three things occur simultaneously: accommodation of the lens, constriction of the pupils, and convergence of the eyeballs, or looking inward. These three things are controlled by the oculomotor nerve.